What I said, I hope I didn't spoil your appetite in the morning. This talk is supposed to be given by my husband, and the slides are coming, Arpad Pusztai. Unfortunately, Arpad had a stroke on the 8th of March, and he's still in hospital, so he can't give that talk, which constrains me to some extent, because I am one of those independent government scientists I am going to talk about. So there are certain topics I cannot tell anything about, only what Arpad has written on. So the title of the talk is Independent Science is a Myth Without Legal and Social Protection of the Scientist, and that's a personal account from him. Well, it's not up to me to speak up, but I hope that they can hear that you can't hear me. Science originates from our natural curiosity to find out about the world and about ourselves. It must be objective and mustn't be driven by profit, which is a very important consideration, or material gain, or wanting to have some glory. In modern time, only the so-called gentleman scientists, which worked at the beginning of the 20th century, and the best example is the double Nobel Prize winner Sanger, who came closest to the definition of a properly independent scientist. Those people had their own individual means, they were usually rich, and they could afford to pursue whatever inquiry they wanted to make on whatever field.
Sorry, 1 second.
Is that better? Excellent. So what I want to tell you that while today most no? Is that all right? Is that all right? Sorry for the technical hitch. So, that's it.
Got it? It's because you've got cute little ears. Yeah.
Thank you. So most scientists are no free agent, and there are two types of scientists, and some of them work for industry, some of them for research institutions or universities. But with the exponential growth of research and research expenditure, research really became a business and too expensive to the government, or for the government to pay for. Therefore, government welcomes the involvement of industry and other sponsors. And this one, unfortunately, started with Mrs. Thatcher. If there is no proper government funding, of course, transnational large companies come in and try to fund most of the research, first of all, in biotechnology and pharmacology. Under these conditions, of course, academic freedom is little more than an illusion. And it's perceived to be in the interest of most government not to pay for independent research because it's expensive. And anyhow, biotechnology research is rapidly becoming a quasi-religious crusade promoted by zealousness, tolerating no dissent, and that's what we did, we dissented. So anyone standing in the way of biotechnology and its targets is declared to be a Luddite who wants to stop science, stop scientific development and progress. As I mentioned, there are two types of scientists, and depending where do they work, they have got different circumstances and conditions to work with. Those scientists who work for the big companies, multinationals, they get very good salaries, but they are hired by the industry for a particular job with a target, which is restricted in its scope, in its objective, and it's carried out under strict supervision. So they can't do anything else, but they were told to target this, achieve this. If they've done that, that's it. If they find anything interesting in the process, they can't pursue that. Of course, all the research results belongs to the company, and they have absolutely no right to discuss it without permission from the company. And of course, they can't publish them either without permission. And if something very interesting and the company might hope to patent it, then that permission can be withheld for 5 or for 10 years or even longer. Actually, what the company says that patenting something, that's an option. But a patent can be broken. And there are people who are officially making patents. We were one of those people. So you get a job to find out how you can go around and achieve the same aim, the same goal with other methods. So the safest thing is keep it quiet, keep it totally to themselves without anybody knowing about it. And that will come later in the talk how important it is. Now, if you think that the situation of a research scientist who is called an independent government advisor or scientist or working for university is better, you are wrong. University or government scientists is in the same situation because when they are hired, they must sign a contract which restricts their freedom. Of course, they can get research money for certain topics. They can pursue only those. If they get something interesting, it will be, of course, wanted to be patented by the institute. And if they want to publish anything, they have to have the written permission of, in case of research institutions, the director, in case of universities, from the dean or the chancellor. And it's very unlikely that if an idea or a result comes up which can make money for the institution or the university, that they will get the permission to publish. And of course, again, with the patent, it can be delayed for up to 5 years. So the question is that how the government or the research institutions and the universities can control the scientist. And it's done by several methods. One is legally binding restrictive contracts. So when the person joins a research institute, they have to sign a contract that all their product, all their result will belong to the institution or to the university. Of course, allocation of research funding by the state and by industrial agencies is again a way of controlling science because they set out certain priorities and money is given only for those. So no research is supported out with their objections. So if you find something interesting, want to pursue it, no way, you can't get the money. Of course, there is an institutional control over all the publication and all the results, vetoing those in conflict with accepted dogmas of the day. And that, again, can be done by contracts and by refusing the written permission. And there is a whistleblower who wants to give away all the nasties which he or she came across. There is a concerted action by the establishment to destroy their credibility. And it's exactly what has happened to Arpad. And I have to tell you that with me and another 20 people, because that research was done by a group of 20. You will never hear anything about them. Arpad put his head over the parapet and he was shot at. So that's a personal account from him, and I'm going to read what he wrote. In 1995, the Scottish Office of Agriculture, Environment and Fisheries Department commissioned a 3-year multi-center research program. Arpad was the coordinator of that, and the main task was to establish credible testing methods for identifying possible human, animal, and environmental hazards of genetically modified foodstuff. So it was not to release the GM potatoes we worked with. That's what people are talking about. Our job was to set up a testing method, the methodology, which can be used for any crop to see whether they are safe or not for humans and animals, whether they will damage the environment or not, and that's for the UK and the European regulatory authorities to be used in risk assessment. As a model, since we couldn't get any seed from any multinational company with the proper control, and of course for GM, you have to have the parent seed which has been converted to GM to be able to compare and see whether there are any deleterious effects or not. You couldn't get that, and you still cannot get them because the companies, I am afraid, they are afraid. If you come up with anything they don't like, what can they do? If they give you any seed samples, you have to sign a contract that all the research results belong to them. You can't talk about them, you can't publish them. So if you find anything which is not agreeing with anybody's health or with the environment's interest, you can't talk about it. Of course, under these conditions, we couldn't sign a contract. We managed to find a small Cambridge company using exactly the same method as Monsanto and other biotechnology companies using for genetic modification. And as I mentioned, we used potatoes as a model produced by that company. And we used that lovely flower, the Rhodops, because the Rhodop is not touched by any predators. It always comes out, the bulb is intact, because there is a so-called lectin in it. It's a protein with insecticidal properties. And we studied that Rhodop and the insecticidal lectin in it for a long time, and we considered it safe. So unfortunately, when we used the potatoes transformed with the gene from this natural insecticidal lectin from the Rhodop, we find quite a few unpleasant surprises. It's true that the GM potatoes has increased resistance to aphids and nematodes, but in addition to protecting the potatoes from these pests, they also could harm beneficial insects preying on aphids like the ladybirds and other non-target insects. And they also damage the gut and other organs of the rats when they were fed to them. And of course, we use rats as a model for mammals and for humans. The question was that what a conscious scientist could do with findings like this when they are so important and so far-reaching in terms of human health, particularly when we learn that the UK public is, without knowing it, is exposed to GM maize, GM tomatoes, and GM soya by the time for 2 years. And those crops hasn't been tested with the strict controls and methodology what we used for our potatoes. And we knew that because our director one morning turned up in the office, shoved 11 volumes of that size on our desk and said that I have to advise the minister whether these crops are good or not, can we release them or not, can you look them through? Now you can imagine the task. With Arpad, we sat down, went through the dossiers and couldn't find any proper animal experimentation. So we were sure that those crops, which were already in our food in the shops, haven't been tested with the same rigor. So there were two choices. Either we keep quiet, get the research money, and carry on with our work, or publicly indicate our concern over the possible health and environmental effect of the untested GM food. And Arpad decided to choose the latter, and it was very easy because Channel 4 approached the Institute that they would like to make an interview at the Institute. And with the RALS agreement, Arpad gave a short interview. It was 120 seconds, 11 sentences, and highlighted our concerns over GM, but without publishing any data. So much so that he wouldn't even know or say what sort of gene we used. And that was a mistake, as we figured out later, because the Institute decided to capitalize on the great public acclaim and the popularity after the broadcast, and without consulting us, released inaccurate information about the genes and the organism we tested. After it appeared that the commercialization of any GM crop food might suffer as a result of the interview, of course, the Institute and us has been attacked and silenced. And the reason was given that we misled the public with inaccurate information that, in fact, was released by the Institute. And they made Arpad to retire, which is very interesting because in the civil service, you have to retire at the age of 60. By that time, Arpad was 68, retired for 8 years, and the Institute just kept him on because he brought in so much money with his research and fame. So the interview was broadcasted, but there were consequences. The media publicity after the interview brought home to people that they were guinea pigs in a gigantic GM testing experiment. And Arpad said it, that the guinea pigs should be found in the laboratory and not people used as guinea pigs. And of course, supermarket sales of GM food plummeted. There were rooftop protests at supermarkets and all sorts of pulling up experimental crops, as I'm sure you might remember. And of course, the government panicked and they started to pressurize the institute. Now, how it was done, I can't exactly tell, but gossip says that Monsanto phoned Clinton, Clinton phoned Blair, Blair phoned the director, Professor James, and after that, Arpad was silenced. It's not the worst part. The worst part was that his scientific integrity was really questioned. It was told that we didn't do any GM experiment, thus he just fabricated the result. He was a cheat, he was a fraud, everything but a killer. He stole other people's data, came public without knowing what he was talking about. He couldn't understand other people's data whom he was presenting, and all sorts of nasties. So what are the methods which are generally used for whistleblowers? And he wasn't the only one. First of all, one has to silence the whistleblower scientist by invoking his or her legally binding contract not to disclose any information without permission. So if the director refuses or the dean the permission, no data can come out at all. The second step is confiscate all the research result and stop all the work. Because if something nasty is coming out, the worms are crowing off the food, then the only thing to stop the research. And use all means to influence or to prevent the whistleblower to find another job. That's one of the reasons. Someone asked me, why don't I work in the UK? I don't think I will ever get another job here in this country. The terms of the contract of the civil service scientist will prevent all this. And the way it is done, that the employee mustn't disclose information or data obtained in the course of duty without the written permission of their director. So no written permission is given, no data can be disclosed in any form. Communication based on these must be submitted to the director before publishing, even if one wants to publish, even after the employment ceased. And an example of that I was, of course, co-author of the Lancet paper, but my name hasn't appeared on it because if it would have been, I was still working at the RAD. The RAD could prevent the publication. So it was much easier for me to be left out as a famous author, but the publication should go ahead and people could learn about the data. And of course, civil action could be raised against the employee who breaches confidentiality, and we have been threatened often enough with that the institute can claim undisclosed amount of damages for ruining their fame. Divulgence of confidential information, even after leaving the institute, will still render the employee liable to legal action. I hope that my talk today won't evoke that response. The other possibility, of course, is to constrain inappropriate future actions by the employee and enable the employer to recoup any so-called financial damages. Generally, I would say that even in MI5, I think there is a 25 years secrecy agreement. In civil service, there is none. If I happen to live to the age of 75, I retired for 25 years from my institution and still wouldn't be able to talk about what has happened there, in addition to what has been already published in the press. So all past and future action of the employees will be appraised in the light of these terms. And whenever you read about science, scientists and statesmen, you have to keep that in mind. And of course, there are personal consequences for lack of protection, and it's usually loss of job, earning, and the status in society and in science. With this comes stress-related illnesses. In Arpad's case, that was high blood pressure, his first heart attack. I'm sure that his stroke and all the other health problems all came from that. Loss of freedom and privacy. the Institute monitored all our phone calls, all our emails. For the first 2 weeks, actually, our phone calls were diverted and our telephone was tapped. Our private mail at home opened. It wasn't secret, you could get it opened and resealed. So the deprival of the fundamental human right of self-defense, that was the worst thing, because Arpad couldn't say anything to protect himself from all these accusations for at least half a year. And I can safely say that that was the hardest time of his life. Of course, there is isolation from science and society as a result, because people say that maybe he did something wrong if he won't come out and say that I'm right and I did this or I did that, but he couldn't. But of course, there are consequences for society if there is no legal protection, because the information flow is blocked. And people are deprived of their democratic right to know what's going on with their food, with their environment, and so on. And the debate, if there is any, becomes one-sided, unbalanced, and depriving people from the right to influence their future and the decision by knowing actually the true facts. And that's the way to manipulate public opinion, and we can see plenty of that nowadays. And of course, there is a loss of democratic rights of calling all the wrongdoers to account. Because if there is no causal relationship between effect and the end results, how can you say that the person or the company who caused this is responsible for the damages? There is no way to do that. And as a result, public health can be in jeopardy. So there again, what are the methods to destroy the whistleblower's credibility? The first step is set up a committee of senior scientists to investigate this vendor, and that has been done by the RIOT. It's a nutritional institute. They set up a 4-person committee to judge the nutritional data, and not a single nutritionist was on that panel. There was an immunologist, a microbiologist, a molecular biologist, and I don't remember who the first person was, but no nutritionist there. And of course, then the job of the committee to release the damning finding of the report to the press, but actually giving the actual data, so they wouldn't know what has been criticized and how. And of course, to repeat that at national level and recruit senior scientists of the establishment who can tell the media that why should they not believe the whistleblower's result. And that has been done, involving the Royal Society, the Nuffield Foundation, and I don't know who else. And at the end, the head of the Royal Society, then Aaron Klug, stood up and said that what a rubbish experiment, the control and the experimental animals wouldn't get the same diet with the same protein content. Now, he is a clever man. If he would have read the papers, the first sentence in it, that all the animals were exposed to the same protein content in the diet. But that's how it is being done. And of course, they have to prove that the scientist is untrustworthy because he or she is unreliable. In case of Arpad, it's too old and confused. Of course, they are not properly qualified to carry out their job. That's very interesting because, of course, you can't get a 1.6 million pounds grant nowadays without actually going through a pre-review process. And there was something like 27 competing projects, and we won. So our project went through, it has been evaluated in advance, they checked the methodology, they checked everything. At the end, we weren't properly qualified, we didn't know how to use the right methodology, and we have ulterior motives, and we wanted to seek publicity and fame, which of course wasn't true. So they have to also prove that the scientist is dishonest, and that has happened to Arpad because he misappropriated others' data. That was told. I have heard Professor James on Radio 4 in one morning standing up and saying that Arpad steals somebody's data, went public with them without knowing what he is interpreting. And he wasn't qualified to know that now. As he, as the head of research group, all the data went to him. He didn't have to steal anything because he had it all. And of course, he could interpret the data because those people who produced it, most of the case could not. And of course, because he or she falsified some of the crucial data, which again was told but has never happened. Because he or she intended and fabricated data. And anyhow, the results can't be trusted because the design of the experiment was flawed, and I've told you about the pre-review anyhow, and the methods were unsatisfactory or biased. The methods has been checked, of course, in advance. Inappropriate controls were used. We didn't use the right statistics. The conclusions weren't supported by the results. That's, I am afraid, all rubbish. But to tell people who haven't seen any of the results, any of the methods, any of the conclusions, one can say whatever they want. The results cannot be trusted either because they are unpublished or published in a journal of low standing. In the previous talk before me, the Lancet has been published. Lancet is the second best medical journal on earth. The results were published in Lancet, so that straight away became a journal of low scientific standing. Or there are better but unspecified data which contradict the results. We were told that in China someone did some experiment with GM tomatoes which contradict our data. So how can one compare potatoes with tomatoes? Totally different genes. So the whole thing was very suspicious. Anyhow, the results are not statistically significant or obtained by using poor statistics, and they are underrepresentative. We were told that therefore the study needs to be repeated with a more refined design and methodology with experts. 10 years has passed, and those experiments or similar one has never ever been produced either in this country or anywhere else. But for the independence of science, this is a very dangerous situation. Accepting money from the biotechnology industry, which has aggressively set out to dominate life and the society, makes scientists and science the servant of multinational companies. I wouldn't say servants, I would really say prostitutes, because they do something for money. As they are most of the time outside democratic controls, I mean the multinational company, and they have no accountability, these big corporations may threaten our democratic institution. Unfortunately, we know they corrupt our politicians. And if you remember back to Ian Gibson, who was head of that inquiry, Arpad appeared in front. Now we know that he strictly represented the interest of the biotechnology industry, and he admitted as much. And they corrupt civil servants and scientists as well. As I mentioned, keeping the public in the dark is the main and center strategy used by the industry, particularly by the GE biotechnology industry. And the reason for that, if there are patents, one can get around them. But if no one knows anything about it, it's very difficult to know what's going on. And I am afraid we don't really know what is going on with GM food. There's a total lack of transparency, and several methods are used by the industry to achieve this secrecy. One is declaring research results as confidential business information. Now, that's a very interesting story because we have seen a few dossiers and even the reference lists, there is absolutely nothing secret about, has been stamped by confidential business information. Arpad reviewed one of the GM dossiers for the German government. And when Monsanto learned about that, took the German government to court, that they allowed someone to get into their secret confidential information, although he signed confidentiality. He couldn't talk about his data, and he didn't. But when Greenpeace learned about it, that he has been in a sense silent second time, they wanted to go open, and they also went to court. And as a result, the German court decided that animal feeding data cannot be kept confidential information, especially if we are all eating the same stuff. So that's a very dodgy business. But of course, if one can withhold publication of unfavorable data, that's again a way to change and manipulate public opinion and make things not very transparable. There is a campaign of disinformation. And also what is going on is really falsifying research data by the industry and generating doubts about the validity of uncomfortable result. And I am not just saying it, but I will show you an example at the end of the talk about this. Of course, if there is no transparency in research, then with irreversible technologies such as genetic engineering, the dependence on research on industrial funding will lead to several negative effects, like the loss of accountability, which I already mentioned, can bring harm to human and animal health, can induce environmental effects which are irreversible, and without exchange of data, of course, without discussion between sciences, scientific progress is impossible and science will die. And there is an increase in all forms of scientific fraud because no one can check the data, repeat the experiment to see whether they are true or not. And then I would mention some examples as the major harm done to secrecy. First of all is the tobacco industry, hiding data showing that smoking was a main factor in lung cancer, heart and circulatory disease, prevented society to take an early preventive action. Other thing was hiding the result of the BSc studies showing that possibility of spread of disease from cow to humans and from cow to the offspring, which led to deaths of over 100 people and led to destroying several thousand animals. And the funny thing is, again, that as researchers, we already knew that this spread is possible from mother to offspring. And I heard on television to stand up the government scientists saying that it's impossible. And I'm sure as we knew the data, he knew it too. The other thing, of course, hiding the evidence that genetic engineering had a role in the death of maiming the people who took GM tryptophan supplement, and the company who produced that destroyed the GE bacteria used in the production of tryptophan, that they wouldn't be held responsible. And I don't know how much do you know about it. There was a court case in the US. Over 100 people went for damages to the company called Shovadenko. That company hasn't got enough money, so all the biotech companies put together a budget which was distributed between the claimants, but they had to sign a piece of paper that they never ever talk about their illnesses, about the compensation they received, or what was going on in court, so that we don't know. And the reason for lack of transparency is to deny the possibility of knowing and the know-how for the competitors, which is not very important because they are sharing most of the patents anyhow. But it's also to make it too difficult to relate an environmental or health calamity to a definite cause in an absence of a well-authenticated research data and evidence. And that's more important. And to escape responsibility for the harm caused by these companies and avoid to pay for the damages. The damage caused by the globalized GM biotechnical industry is several fold. One is blocking research on the possible irreversible health, environment, and economic risk due to the spread of potentially hazardous GM crops. The other is blocking research into the destruction of biodiversity, soil quality, and food security. Growing dependence on the farmers on monopolies and seed provided by the monopolies. And of course, withholding funding from further research into the new genetic techniques which might be better or might be able to produce better stuff if there is such a GMO. Also, the destruction of independent scientists by the financial and political power of the GM industry is a tremendous damage, and there are several people who went through that process. Effectively, there is no GM regulation in the USA. Absolutely nothing. The company just has to notify FDA that within 6 weeks they are going to release a new GMO. And as a result, there is no safety assessment. And if there is anything, it's based on the poorly defined and equally legally not binding concept of substantial equivalence. I am going to tell you in a minute what substantial equivalence is, but as again, the risk assessment and the result of it is confidential business information that is not available for the public, and independent scientists can't scrutinize those data. And of course, what companies do, the biotech companies, they're using the power of WTO and the GM biotech companies to force other nations to accept their GM products And the best example may be Iraq, where the new constitution introduced the seed list, so only those seeds which are on the seed list can be cultivated and sold. And of course, on the seed list, only American seeds and several GM wheat varieties were on. And that's not very good for the people there. So let's talk a little bit about substantial equivalence and what sort of dangers GM plants represent. The first question is, how can the plant be novel and the same at the same time? The plant should be novel because that's the only way to patent it, and for that you have to insert a new gene. But if you inserted a new gene, it's true the plant is different, but it should be the same, because if it's the same, you don't have to safety test it. So that's a very interesting situation, because substantial equivalence declares that if the major components of a GM plant, such as protein or ash or vitamins or minerals, are within an acceptable range, and the keyword is the acceptable range of a non-modified plant, the parent plant, then they are substantially equivalent. For instance, if I've got a potato, which has been genetically modified, and they have got the same protein content, the same vitamin content, they are substantially equivalent. If this plant, the original, was safe to eat, then the GM plant, because it has got the same chemical composition, it's also safe to eat. So there is no need to test for anything. And since the traditional crops all have got this history of safe use, practically no GM crops has to be tested, and they are all safe to eat anyhow. And that's the view, unfortunately, not just by the American regulators, but by the EU as well. So all the crops at the market at present has been released on that idea of the substantial equivalence. So who has introduced this idea? Of course, the GM companies and the regulators. Why? To make big profit by selling these seeds. Because just imagine that our program costed 1.6 million to the British taxpayer. How many carrots or potatoes do you have to sell that you would test it properly to get 1.6 million pounds for the test? And of course, by not testing, you can spare plenty of money. And now I would like to talk about some individual GM crops. The very first one was on the market was the so-called favor saver tomato, which was the product of an antisense technology, which means that they didn't introduce a new gene, they said, but put one back in a reverse position. Anyhow, it was claimed that the insertion of that gene caused no changes in fruit composition of the tomatoes. However, daily dosing of a normally fed rat with the GM tomato to led some potentially serious health problem. And that was a research project which was commissioned by FDA, the Food and Drug Administration of the US, responsible for that. And they did the following experiment. They fed 20 male rats with non-transgenic tomatoes, and they couldn't find any histological alterations in their stomach. They fed 20 females with non-transgenic tomatoes, and there was no effect whatsoever. When they fed transgenic tomatoes to 20 male rats, there was no effect. And it's very interesting then when they treated the females with the transgenic tomatoes, they could find foal animals which had stomach erosion. To turn it or translate it to proper term, it's bleeding of the stomach, which can be quite dangerous. So FDA was slightly worried, collected the slides and sent it to another laboratory when they analyzed it. And they said that it's not in 4 rats they can see these bleedings, but in 7. After that, FDA decided that's a very dangerous business to do this animal test and said that there is no need for animal testing in the future for any GM crops whatsoever. But what are the implications of this study? In humans, stomach bleeding can be life-threatening, particularly in the elderly, or I'm sure you know when you take non-steroidal anti-inflammatory drugs like aspirin, you can have stomach bleeding very easily. Combining the two effects can be deadly. And if it is so, it was shown by this animal experiment, because 7 out of the 40 rats eating the GM tomatoes, we don't know whether they were male or female, died. And what they do, in the research place where they carried out the experiments. They took out the dead animals and replaced them with live ones. Now, a proper scientist in an experiment will never do that. The next one is GM potatoes. I've already talked a little about our experiments, but we weren't the first one, or not the only one, because in 1998, when the calamity has happened to us, a paper appeared with BT potatoes, and it showed that it caused potentially harmful disruption of the surface of the gut structure by swelling and inducing cell growth, producing more cells in the ileum. And also these effects demonstrated that the BT toxin, which we were told that it's harmless because it degrades in the gut, actually it survives and could bind and has a strong effect on gut metabolism. There again, data with our GNA tomatoes. Feeding rats with these GNA potatoes induced growth in their stomach, the small and large intestine, and I'm going to show you a histological slide in a moment. It induced lymphocyte infiltration in the gut. The gut behaved if there would have been a microbial or bacterial infection. The body's immune response slowed down tremendously. And the biggest problem of ours was that we realized that these effects due to the transgenic process converting the genome to a transgene and not the transgene itself. So whatever transgene they use by this method will have this effect, and that was our downfall because the companies realized how dangerous it is, and it's the end of all GM products. And these are the histology side I promised. There's the same magnification. Here you can see the control potato and the GM fed potato stomach histology. You don't have to be a pathologist to see the changes, the increased cell number and the proliferation, which of course can very easily lead to stomach cancer. So to summarize the finding of our GM potato study, the composition of the GM potato lines weren't substantially equivalent to the parents, or as we used two different GM lines, neither to each other. So the substantial equivalence wasn't done. As the protein content and nutritional value of one of the GM potatoes was reduced, the growth of young rats on this GM potato diet and their feed conversion efficiency was significantly less than the control potato-fed animals. The GM potato feeding had a mitogenic effect on the gut, as I showed you, in young rats, and interfered with organ development and compared with the isoproteinic, isocaloric diet, so with diet where there had exactly the same protein and nutrient and calorie content, these changes were pretty obvious. GM potatoes also increased the gut immune responsiveness and reduced the humoral general immune responsiveness of the body. That was damning enough. But let's see what's going on with other crops which are on our shelves. Hopefully not so much in the UK, but in Europe, the GM maize. It became obvious from experiment in the Philippines when farmers started to grow GM maize. It was Monsanto 810 with the BT toxin in it, that they developed allergenic symptoms. And it was a very interesting case because when the maize started to pollinate and the pollen spread in the air, those people living nearby became really sick and showed all signs of allergenic diseases. But that time coincided with the national holidays when families visit each other, and those workers who left the place where the GM maize was grown went away. Their symptoms disappeared, but those people who came to visit people living near to the GM field, they developed immunology problems. So one of the Christian medical organizations working nearby collected blood samples from these people, and they could detect specific immunoglobulins against the BT toxin in the blood of these people. And that's a very serious proof, being ignored, of course, by the entire biotech industry. Anyhow, allergenicity, it appears that it's the Achilles heel of GM crops, because there are several cases of GM crops being allergenic. One well-publicized case is by Prescott et al., when they took the alpha amylase inhibitor. It's an enzyme inhibitor which prevents carbohydrate degradation in the stomach. And that natural inhibitor is present in beans. And it's not allergenic. So they took that inhibitor, which is a very good insecticidal protein, and transferred it to peas, which is a closely related species. And as soon as it has been transferred to peas, it became allergenic. And that's quite a serious problem. Now, then, the transgenic transfer of the donor plant of a gene which is coding for a non-allergenic protein, even when it's transferred to a closely related species like beans and peas, led to a synthesis of a new type of protein with altered structure and altered immunogenicity, so it became allergenic. The other example is the soybean. I don't know whether you do. In New York, the Allergicity Institute always gives out the data on allergy in the entire country. And there was a steady increase of soy allergy, but from 1997-98, it started to go up steep. And they realized that it's because of the introduction of GM soya. At that time, people didn't know that they are exposed to it. And the reason for it, that GM soya contained an IgE, an immunogenic protein, which is not present in the non-GM soya, because it has got different properties. And that's, again, published. It's not our data, so I'm not trying to make a case for ourselves. The other problem is GM DNA and proteins in milk. And just remember this slide when we are talking about what the FDA scientists told at the end of my talk. So we will come back to this, that although the source is disputed, whether it's from partially digested GM feed or contamination from the air or feces, because of course the data has been explained away several times, but GM DNA was found in milk by two set of research groups. And in one case, is the cry-1-way toxin was detected in milk from Bt maize-fed cows. So it appears that the GM protein from the feed can transfer to the cows, from the cows to the milk, and I am afraid to us too. As I mentioned, the excuse is always that Bt toxin is not binding to the gut of mammals. It degrades, but there is quite definite evidence in the literature that it binds to the mouse jejunum. In vitro, the protoxin binds to fixed gut sections. The mouse gut contains at least 6 different proteins which can bind the toxin. And Bt toxin is an immunogenic and an allergenic protein, and it's also an adjuvant. I don't know whether you know what an adjuvant is, but when you get an inoculation against any of the diseases, they put it, usually cholera or diphtheria toxin in small quantities to boost your immune system, and that's called an adjuvant effect. So when someone eats Bt toxin and eats some other allergenic proteins, it will become more allergenic, or if it was non-allergenic, it can become allergenic if it's eaten together. That's the adjuvant property, and it's well publicized as well. There is well-described effect of GM soya on murine liver and the pancreas. GM soya affects the structure of the liver cells, changing the size of the nuclei, making them bigger and irregular. And it is an indicator of increased metabolic rate. Digestive enzyme synthesis in the pancreas was also reduced, and soya consumption was linked to pancreatic adenoma, pancreatic cancer in rats. GM crop herbicide safety, that's another issue, because of course these GM crops are sprayed with glyphosate, and the formulation can delay the cell entry to the M phase at an early stage of development. In other words, if embryo is being formed, there is interference with the mitosis. The glyphosate will delay the hatching of C-Uchin eggs, will increase liver damage in gut. It's toxic to the human placental cells at a very low level. It's an endocrine disruptor, causing pregnancy problems and abortion and skin irritation and several other things. Having said that, there was this independent evaluation of the Monsanto 863 GMA study which Arpad did, which showed the growth of kidneys and livers in rats fed the GM material. And that maze was significantly different from the controls. And the similar data were published by Serolini 2 years ago. And it is impossible to expect at reduced growth rate histological changes in important organs and other parameters which change in blood, like lymphocytes, reticulocytes, the glucose level in rats are incidental and due only to biological variation. That's the way how they try to explain away the differences. It has no biological significance, it's biological variation. Up till now, there were 2 human studies carried out here in the UK. One is known as the so-called Newcastle experiment. And the aim of that experiment was to see whether the transgene from GM soya, you can see the cell, the grown GM soya, when it's made up as a milkshake fed to people, it can be converted and transferred to their gut bacteria. Now for that, they used ileostomy patients, people whose large intestine has been taken out and the end of the small intestine, the end of the digestive tract is sewn out on their stomach wall. There is a bag joined to that, and it's very easy to sample that. So that's why they use 7 of these volunteers. And I'm going to show you the data. Out of the 7, the entire full length of the transgene from all the parts, from the gene switch to the marker gene, was present after 4 translocation in the GM bacteria of this patient. And the only reason that the 7th is not on the slide, because they always put in a good experiment a marker, and they couldn't get that marker back 100%. So the data for that person has been left out of the study. So this study clearly showed that GM soya or any other GM crop with the transgene incorporated into it can get through to the human gut and can be incorporated into the gut bacteria, and I'm afraid in several cases into the gut cells themselves. That hasn't been published, but it's result produced in Tromsø in Norway. So the question is that are we really guinea pigs in GM food experiment? And I am afraid we are. It's a gigantic experiment with humans. And there is another trial which just went on lately in the States, when 24 children took part in a clinical trial to try the golden rice on them. I don't know whether you know and heard about golden rice. That produces vitamin A provitamin in some quantity in the rice. So if you are fed with that, you won't have any vitamin A deficiency, which leads to blindness. The problem with that rice is that there is no full information about the plant's genetic makeup. It hasn't been approved anywhere on Earth, and they have never tested this rice on animals. So the first port of call, so to say, is the human and especially children's. And as far as I know, it's very, very difficult to get permission to do experiments with children. So we think that it's a GM variety with a very unique danger. But the most upsetting thing is the effect of GM on reproduction. And if you remember back to my talk in the morning, I told you that when a female of a species, including humans, are born, in her ovaries, the eggs for the next generation is already there. So there's a 2-generation delay with everything. Please keep that in mind when I'm going to talk about the next set of experiments. The first experiment was done by Ermakova in the Institute of Academy of Russia. It's a very respectable institute, and I have seen the data. I couldn't find any fault with them. So she wanted to know what is the effect of GM soya and non-GM soya in comparison on reproduction in rats. And what she did, she fed the animals 2 weeks before mating, during pregnancy and lactation, with either GM or non-GM soya. And I am going to show you the result of the number of props that grows as it was followed. You don't have to see any of the details. The most important thing that on the third column there is the red marks, the red numbers. On control, only 3 of the pups died. On GM soya, 25. And on traditional soya, only 3. And also, the weight of the animals were smaller, but it's easier to see the mortality rate on a graph like that. And the differences are quite significant. And if you can see the picture of the pups, guess which one is the GM fed, the smaller one, at the age of 19 days old. So these experimental results are quite frightening. So the conclusions of the Ermakova study was that she said that the litters produced by the pregnant and suckling female rats fed the RR soya had significantly higher number of dead pups, as you could see. The weight of the pup who remained alive were much less. And the question one has to pose that if we are feeding our pigs or chicken or other animals with the same GM soya, what will happen to livestock? And in January, I just heard that pig farmers in the US took Monsanto to court because several of the sows became infertile and some of the sows gave birth to bags of water. That hasn't been published, so that's just, you can take it as gossip, but the court case is definitely on. With GM soy, there is another observation which has been published in a scientific journal, and that's that the testicular cells had altered structure and function. And there again, you don't have to be a histologist to see the difference between the control and GM-fed testicles. So if you affect the reproductive function of the females and the males as well, what will the future bring? The other study on reproduction was commissioned by the Austrian government, the Ministry of Health and Environment. And they asked a lady who is well known about her reproductive experiments, Alberta Velmirov, who did the comparison between organic and conventional. And they designed an experiment where they fed animals with only 1/3 of the diet is being either GM maize or the parent line, or specialized types of Austrian maize, because they realized that the parent line, which is supposed to be GM-free, has been contaminated at the area where it was grown. Because, of course, you have to grow the GM and the non-GM line next to each other under identical conditions. The only thing they didn't count on was cross-contamination, which has happened. So they had to use a third line. Anyhow, they did 3 experiments, and they are all very interesting. One, what they call the multi-generation study, where they followed the reproduction ability and outcome over 4 generations, so great-grandparents, grandparents, parents, and children. The other was what they call the reproductive assessment by continuous breeding, when they used the same breeding pairs, the same parents, to produce 4 pregnancies and check the number of pups and their growth. And the third one was a lifetime study. But they used a special type of mice which dies from leukemia anyhow. So with the lifelong study, they couldn't find any differences on the 3 diet. But when they did the reproductive assessment study by continuous breeding, what they find that on GM, the number of infertile female increased by pregnancies, and by the 4th, this became significant. The number of offspring was always lower by the 3rd and 4th pregnancies. Remember, there's a 2 pregnancy or 2 generation delay at least. More offspring were born on the ISO diet, 1,035, compared with the GM, 844. That would be a very nice way to decrease the population on Earth, but I don't think that would be my chosen method. The number of pups per mother was also less on the GM diet, and of course the weight of the 4th offspring was significantly lower on the GM diet. With the multi-generation study, when they followed it through for 4 generations, from the F0 generation, all animals kept on GM were the same till the offspring was born. There was no difference in food intake, body mass, no number of offspring and their mass up to the 4th generation. And I'm going to show you the data because the rest is very specific, more for scientists. What they find that With the exception of the F2 generation, there was always more pups born on the ISO diet than on GM. It's pretty obvious. Over the generation, more pups died on GM, 14%, compared with the ISO diet, the parent lane, where it was only 6%. And for the first 3 generations, the litter mass wasn't significantly, but always higher on the control, and it became significant for the 4th generation. And the electron microscopy showed alterations in the structure of hepatocytes, lymphocytes, and the acinar cells of the pancreas. So whatever you have seen with other crops, it was coming back in that study. To summarize this experiment, on GMAs, there was no effect on lifespan, but significantly reduced the reproductive ability of the animals. The morphology of the liver and pancreas and spleen was significantly altered. The gene expression of several proteins was influenced. And there again, we are coming back to the same thing, that this type of experiments are not required by the regulators at all. So, if we are there eating that food, let's go back for a moment to see that what is the state of science and scientists. You remember my introduction, what I did say, what happens to those who whistleblow or go against the accepted dogmas. There is certainly a damage to science by industrial and political interference, and in the States, the Union of Concerned Scientists prepared a report on the so-called disinformation syndrome, which afflicts, according to them, all government scientists. They made their survey and find out that 1 in 5 scientists had been directed to exclude or alter information in a scientific document, which means that 1/5 of the publication was nothing, not the paper is written on. More than half of the 3,247 scientists who has been asked said that commercial interest through political intervention directed them to reverse or withdraw research conclusion. So now we are at about 50% of scientists who falsify or change or keep quiet about their data. Another survey by the Institute of Professionals, Managers and Specialists, they asked 500 people, and 30% of them said that they have been asked to tailor their research conclusion, such as 17% was told to change the result to suit the customer preferred outcome, so those people who paid for the study. 10% was to change if they wanted further contract and further money for research, and 3% was not told not to publish unwelcome results, which means that those data which are deleterious or should warn us, they are not published. The third survey was very interesting. It was the US Fish and Wildlife Service concerning the protection of endangered plants and animal species. And they revealed that 44% of the respondents reported that they have been directed to withhold data which would have indicated that a species of animal or plant needs protection. It means that we don't know really what sort of plants and animals need protecting. 20% reported that they have been instructed to compromise their research and scientific integrity by excluding or altering data or opinions. That's what it's called, simply cheating and falsifying or creating research data. And the most shocking was that 56% of them new cases where commercial interest included or induced reversal or withdrawal of decisions or conclusions through political intervention. So more than half of the results are influenced by politicians, by lawmakers, by regulators. And it's quite frightening because it leads to the conclusion that you can't trust anybody's data. Of course, it causes damage to everyone. And the most shocking example is the NIH, that's the National Institute of Health in the US, when 103 government scientists has been asked to reveal whether they have ever violated any ethical rules or collaborated with pharmaceutical companies without admitting to it. And out of the 103, 44 said that they violated those rules. 9 said that may have violated criminal laws. And the most upsetting thing that there were 274 complaints to the NHS in just 1 year in 2004. But because they have not got enough people, they could just examine 23, less than 10% of all the complaints. So one can say that our health is absolutely not safe because we don't know what we are dealing with. And as far as biotechnology is involved in the whole process, there were all early warning by the scientists working at FDA that there are allergens, toxins, new diseases, and nutritional problems with GM. After all that, FDA declared that GMOs are not different from conventional plants. The agency is not aware of any information showing that foods derived by these new methods differ from other foods in any meaningful or uniform way. And that was a statement in 1992, when no one ever did a single GMO experiment. But secret FDA documents confirmed that the facts contradicted that statement already at that time, because several of the scientists complained or wrote to the head of the agency. They said that residues of plant constituents or toxicant in meat and milk product may pose human food safety concern. Remember back the milk study I showed you? And that was an early warning, ignored. Also at the FDA, one of the compliant officers said that progress of genetic engineering and traditional breeding are different, and according to the technical expert in the agency, they lead to different risks. So substantial equivalent is not the right method to judge their safety. She also said that by trying to force an ultimate conclusion that there is no difference between foods modified by genetic engineering and food modified by traditional breeding, the agency was trying to fit a square peg into a round hole, which is absolutely true. One of the veterinary officers said that animal feeds derived from genetically modified plants present a unique animal and food safety concern. I would urge you to eliminate statements that suggest that the lack of information can be used as evidence for no regulatory concern, because that's what the agency said. And what has happened to the scientific element of the document, that's about another document about GM food. Without a sound scientific base to rest on, this becomes a broad, general, what do I have to do to avoid trouble type of document. It will look like and probably be just a political document. It reads very pro-industry, especially in the arena of unintended effects, which are the cornerstone of GM, as I showed you this morning. So who overruled these scientists? Of course, the head of FDA, who was a former attorney of Monsanto company. And that's very interesting that we are being told that FDA is a responsible agency and they examine the safety of GM food very, very carefully. I would like to read out what the letter said when a company sends in permission to release a GMO. Based on the safety and nutritional assessment you have conducted, I mean the company, it is our understanding that Monsanto has concluded that the crop products derived from this new variety are not materially different in composition, safety, and other relevant parameters from corn currently on the market, and that genetically modified corn does not raise issues that would require pre-market review or approval by FDA. As you are aware, it is Monsanto's responsibility to ensure that food marketed by the firm are safe. So FDA doesn't say that it's safe. It says that the company says it's safe, and it's their responsibility to make sure that it's truly safe. So at the end of this, we all have to remind ourselves and our peers and politicians and also the scientists that any democratic society which suppresses academic freedom and the inventiveness of the individual stifles with its own development and imperils a democratic institution. I think I gave you enough examples of that. So without scientific freedom, there is no progress in science, no safety of health, and no safety of the environment. And we cannot test the research publications. So what is the way forward? The claim of corporate science must be independently verified by publicly funded scientists acting as watchdogs. That has happened to the RALT, our institute, during and after the Second World War. Now it's a non-existing institution. The public must be helped to understand that for independent scientific advice, they have to release scientists from their servitude to business by funding them from the public purse, so the research could be done openly, transparently, and inclusively, that it would benefit for all. And really our message that the absence of evidence of harm never can be taken as evidence for the safety of GM foods or for any other crops or any other products like nanotechnology, particularly if the safety studies are not done. And I am afraid most of the cases are not done. And our motto must be, as new discoveries, products, and novel technologies will afflict us all, we are all entitled to scrutinize the evidence relating to their safety, which of course relates to our health, our environment. And what I was talking about most of the time wasn't our data, and I would like just to show you 6 or 7 slides is full of the references. They are all published data. They are not out of web pages or unconfirmed reports.